All right. Well, thank you very much for having me here to share some thoughts with you. I'm not sure if this is the title I had given you in the beginning, but I sort of rearranged everything and decided to do this talk in a completely different way than what I normally do. So let's see what happens. If anybody's interested in following up afterwards, everything is at this website, papers, software, everything is here. So let's start by just thinking about what this is. So here's an aqueous maze. Here's some kind of autonomous thing navigating this maze. It takes a corner without having to bump into the opposite wall. At this point, it makes a spontaneous decision to turn around and come back. So we might wonder what this is. And you've probably seen these before. This is a Xenobot, and here it is. It's been fixed down, and so the ciliary motion that it usually uses to swim through the water is, you can see it here, pushing the particles past it. And so one thing you might think from looking at it is, if you didn't know what it was, you might think that it's something we found in a pond somewhere, and you might think that it would have an invertebrate genome and so on. But it's actually a Xenobot. It's a biological sort of proto-organism and biorobotics platform that we made out of skin cells in the frog. But I think even more interestingly, it's a kind of vehicle for exploring anatomical morphospace. I see it as a tool, as a kind of telescope or periscope that allows you to peer into another space that we normally don't see at all. And it's a kind of visualization tool for diverse intelligence. And what I'm going to argue even more in this talk is that it's actually just one part of a highly heterogeneous composite entity, which consists of these biological cells, but also an AI, and also at least 4 human scientists. And so I think that's the right way to think about a Xenobot. Actually, is not just this thing, but actually as this composite entity. So what I want to do first is show you a little bit of biology and talk about some concepts, including multiscale competency architecture, the idea that functionality in biology is often hidden. It's often cryptic because we get distracted by the reliable default outcomes. And I make this argument that in biology, every part is basically hacking all of the other parts to shape their behavior. And that gives rise to massive plasticity, virtualization, and the utility of a kind of software hardware lens. One of the basic things in our lab is that we add to the kind of easy way we often think of identifying intelligence in a familiar 3-dimensional space, and so we call this behavior, and we have these medium scale objects moving at medium speeds, and we're very good at recognizing those things as intelligent, apes and birds and octopuses and so on. But in biology, there are all these other spaces. There's a space of gene expression and the space of physiological states, and anatomical morphospace, which is a space of possible physical configurations. Living beings navigate all of these spaces, and we don't have direct perception into this space the way we do in 3-dimensional space, and we're not very good at recognizing intelligence there. But it is ubiquitous in these other spaces, and I can only mention today a few examples. But one of the things that happens with being a navigating agent in one of these spaces is that you can get hacked. That is exploited and trained and communicated with, that changes your behavior. Now, we're starting to be familiar with these kind of hacking examples in behavioral space. So, for example, zombie ant fungus that invades the body of these ants and gets them to do certain behaviors. It goes sort of higher than that. There's this parasite which has a life cycle in rodents that needs to get into a cat, and so what it does is it lowers the risk aversion in these animals, and they end up running around even though they smell cats, and then the cat will eat them. And humans that are infected with this tend to be things like captains of industry, and it really makes it actually makes people, as a whole literature on this, it makes people more willing to take risks, so more entrepreneurial. Lots of people who crash their motorcycle driving too fast for conditions and end up testing positive for this. So this is a very subtle effect on the behavior of a very complex system. And it's not that this parasite knows how to be an entrepreneur, it's that it knows how to hack the host to have these really, really subtle kinds of effects. And so now we can start to think about, what does that look like in these other spaces? So here's an interesting example. This is not Photoshop. I thought it was Photoshop the first time I saw this. This is a fly, which is running a kind of stripped down 2-dimensional ant program on its wings. And what it does, and the reason is that it sort of flaps these wings and it looks like the ants are scurrying around, and it keeps predators away because the predators don't want to deal with these stinging ants. And so this is kind of incredible. And then you think about, well, that's amazing, but even more amazing is the main program here that actually creates the whole fly. So this is quite something, but this is kind of a virtual kind of low resolution kind of example of morphogenesis. The main show is actually here. All of these cells are able to make different kinds of shapes. And one of my favorite examples is this. So you've got an acorn, and you ask yourself, well, what does an oak genome know how to do? Well, it knows how to make this. These cells can make nice flat green things. They do it very reliably. Every leaf looks basically the same, and so there you go. But it turns out that actually these exact cells can also make this, and they can make this. These are galls. They're induced by a parasite, a particular kind of wasp, and there are many other parasites, fungi, and so on, that can do this. And what it does is it reveals the fact that these cells with this exact same genome can actually leave the region of morphospace where all they do is make a flat green thing, and they can travel to this area. They're actually capable of making this. You would never know, right? By looking at how many oak trees can you look at without knowing that actually they're capable of doing this. So this is an example of biology hacking each other and revealing the latent morphospace and the latent physiological space of these cells, right? And so we really have to start thinking about this kind of software hardware distinction. Now, we look at something like this. So here's this frog, Xenopus laevis, that we use all the time, and it's got some kind of body, and it's got some kind of cognitive capacity and behavioral repertoire and so on. And we say, okay, well, here's an organism. And I want to make the argument that we only think this is an organism because we have these really compressed, but pretty tiny lenses through which we see the world. We are very short-lived, very limited observers, and we see this animal, and we think this is the organism. But fortunately, we have synthetic bioengineering and things like that, and robotics, of course. And the Xenobot is one of them. It collapses a much bigger picture onto a scale that we can actually appreciate. In the case of the frog, this is actually the organism. It's this whole thing. It's the evolutionary cycle wrapped around all of the different things in the environment that end up producing the frog. So internally, of course, the genome sets some of the hardware, but also we've got various different degrees of agency in the environment that actually manipulate what ends up happening, from very simple kinds of chemical physical features, simple organisms, and very complex organisms like we bioengineers, right? So there's parasites, conspecifics, and so why is this the organism? Because you need to know all of this to optimize your ability to predict, control, understand, repair, and create, and in the case of more complex beings, to relate to it actually in both a functionally useful and an ethical sense. You need to understand what all the inputs and all the capabilities are. And so as we think about the Xenobot, so what is all this for the Xenobot? What does the broader conceived Xenobot look like? So let's just think about what Xenobots actually are. So pause this. So this is work that was done in collaboration with the University of Vermont. Josh Bongard is our partner for all this work. His student Sam Kriegman did the software, and Doug Blackiston, who's a scientist in my group, did all the biology for this. So what we decided to see was to really test the plasticity of cells and see what else they can do when liberated from their normal constraints. And so we have this frog embryo. We take these cells up here that are they're going to be skin. They're already determined to be epithelium, and we dissociate them and we put them in a little pile here. And now what could they do? Well, they could do many things. They could die, they could spread out, they could form a two-dimensional monolayer, they could do nothing. Instead, what they do is overnight they get together and they coalesce and they form this kind of thing. The flashes you see is calcium signaling, which is one of the things people track in brains, but there are no neurons here. This is just skin. And as a result, you get this. You get a system that is autonomously motile. So it uses these little cilia, these little hairs that normally spread the mucus down the body of the frog. And they have different kinds of motions. They can go in circles, they can patrol back and forth. They have collective behaviors, they have individual behaviors, they have lots of different kinds of behaviors. You can sculpt them in various ways. You can make ones that swim like this, like a fish. Here's Doug's magical donut swimming around. And if you track the calcium, you can do many interesting things. And this is still, the cognitive capacities are still being investigated. What do they sense? What are their preferences? What can they learn? What kind of learning capacity do they have? You can do all sorts of cool information theory on these calcium fluxes, exactly how you would do among regions in the brain, or in fact between two brains on a given task. You can ask what degree of coordination there is. They regenerate. So if you cut them almost in half, they will fold back up. Look at this hinge, 180 degrees. Think about doing like a bicep curl from a straight arm. Like look at the force that's required to fold this thing back up into its new Xenobot shape. And perhaps the most amazing thing so far, and I really think this is just the tip of the iceberg, the most amazing thing that we've seen is Josh and Sam discovered that in a simulation, in an in silico simulation, they discovered that if you sprinkle a bunch of loose material in the vicinity, what they would do is through their actions, they would displace this material in a very interesting particular way. And so part of the deformations and the stimuli that we give these bots come from this virtual world, this AI. It's an evolutionary wrapper around different shapes and different ways to make these bots, and it can make predictions. And Doug, amazingly, Doug can actually make some of these. And so what we found is that if you do provide them with particles that are themselves an agential material, so not just loose, kind of a passive matter, but actually an agential material, what they will do is they run around and they coalesce it into little piles. The little piles, and they make them sort of tighter and tighter. The little piles, because they're an agential material, this is what enables the next phase, which is that it itself becomes the next generation of Xenobots. They mature. And guess what they do? They start to run around and do exactly the same thing. And so that gives you the second generation, the third, and so on. So the reason this works is for the same reason that the Xenobot construction works in the first place, because we worked with an agential material, which are these cells that have an agenda of their own, and they're able to do the same thing. And so this is kinematic self-replication. This is sort of von Neumann's dream. This is some sort of robot that can go around and make copies of itself from materials it finds in the environment. As far as we know, no other creature does this. So we'll get back to some of the implications later. But this to me is a Xenobot. It's not just this. It's not just the actual thing here. The whole cycle that allows these Xenobots to exist involves all of these components. It involves these other intelligences, meaning us in the environment. It involves the cells. It involves the genome of the frog that made some of this hardware. It involves part of the morphospace that is this novel aspect here. It involves the AI actually that was helping us to determine the optimal stimuli and optimal shape. And so this, I think, is a composite entity, and I think we need to really get behind this idea that beings are not just simple physical things that are obvious to human observers. This whole thing is the Xenobot. Okay. And that's kind of strange. We're not used to thinking these highly heterogeneous. I mean, some biologists that study weird symbiotes and so on, they're sort of on their way. But typically we don't think about these really strange composite kinds of entities as a single thing. But we should, because you and I are also collective intelligences, not just ants and bees and bird flocks. All of us are collective intelligences. Why? Because our brains are made of individual cells that used to be organisms themselves. So this is René Descartes. René Descartes really liked the pineal gland because he said that there's only one of them in the brain, unlike all the paired structures. And he thought that for the unified human experience, there should be one structure in the brain that sort of mediates that. But if he had access to good microscopy, he would have noticed that there isn't one of anything anywhere. There's basically in the body, the pineal gland is just a collection of these cells. And in fact, each one of these cells, if you look underneath, it's a collection of this stuff. This is all the stuff that's inside of a single cell. So we are massively sort of multi-scale creatures. This is the kind of thing we're made of, although this is a free-living organism. This is called the lacrimaria. So look at how it's, so there's no brain, there's no nervous system. The whole thing is one cell. It solves all of its local physiological needs, anatomical needs, metabolic needs, all at the level of a single cell. If you're into soft robotics, I mean, look at this design challenge. Look at the amazing control it has over its body. And if you cut off this little thing that looks like its head, it reforms immediately because the whole thing is just cytoplasm. It's one cell. So all of us make this amazing journey from a quiescent oocyte, which is a little blob of chemistry and physics, and we end up as one of these things, and maybe even something like that. So a complex metacognitive mind. And this process is very slow and gradual. There's no magical light flash that turns you from pure physics to cognition. This happens slowly and gradually, and it's by the action of things like this. So it turns out that there are these not just structurally we are a bunch of nested dolls of different levels of organization, but this is a multi-scale competency architecture. Every level solves problems in its own space. So the transcriptional space, physiological space, anatomical space, behavioral space, all, and in the case of humans, social and linguistic spaces. So the body is fundamentally a kind of agential prosthetic. It's a tool that certain components of the cognitive system are using. They're embedded in it, of course. You can't separate them, but it has its own set of agendas. And so it is not just this idea that you've got this brain and then it's driving this sort of Waldo, this body. The body actually is part of the cognition because it is itself. It has various intelligences, meaning problem solving, competencies, goals, and so on. And so I can just show you a few cute examples. This is a planarian, and we'll talk more about this in a moment. This amazing flatworm. And one of the things the planaria have are a pharynx, which is this little tube that it sometimes uses to pick up food, but you don't see it. The pharynx is most of the time is completely quiescent, so you can't see it. But there's this notion that Wilhelm Roux put out well over 100 years ago called the struggle of the parts. And it's the idea that not just the individual cells had to be tamed at the beginning of multicellularity to work together, but it's a constant work in progress to keep all these independent pieces functioning together. And it's sort of one reason why we don't really see, there's another talk that I sometimes give called Why Robots Don't Get Cancer, right? It's because in robotics, we typically don't deal with parts that have their own agendas, but in biology they do. And so here are some pharynxes that have been liberated from this planaria, and this is what they look like. They have a little independent life on their own. They would like to eat this liver, and here they go. Of course, it's just a tube with nothing on the back end, so eventually they sort of just like worm their way through it and come out the other side. But what they're normally, but normally they're kept quite quiescent. This is what happens when you liberate them from those inhibitory signals. And so bigger picture, then we need to start thinking about engineering across the whole spectrum. So this is what we've been doing for thousands of years, engineering with passive materials. Now, and many of you here are doing work on this kind of stuff. We have active matter, we have computational matter, and the tools and techniques are quite different because what this spectrum really is. And it goes through to agential material like cells, which is a material with its own agenda. Not only can it do things, not only can it compute things, but it actually has goal-seeking loops in which case it will actually try to do things. And it has competencies where it will do things despite intervention. That's sort of the point of intelligence in William James's definition of being able to reach the same goal by different means. And so there are really different techniques that you would apply across this whole spectrum. You can't and you shouldn't deal with this kind of material the way you would with this, and vice versa. Both are mistakes. So you need to find the right place. And there have been a couple of different sort of attempts to formalize this scale. So here's Wiener and Rosenbluth's scale back in the 40s. This is one that I've been working on, this axis of persuadability and different ways to interact. And so just to show you again, just the last couple of pieces, some interesting biology. So one thing we would like to do then is to communicate with this collective intelligence of cells. And that means rewriting part of its memory. It means giving it signals not rewiring it like clockwork. So we don't want to do this to living things and have to be in charge of all the micro details. We want to be here, or better yet here, or maybe even here in some really interesting things that are coming down the line. So what we can do is we can look at the memory medium, and that happens to be bioelectrical circuits, which I don't have time to really get into, but we now have ways of reading it the same way that you would read a brain. And you can see that this planarian, this flatworm, has this electrical pattern that tells it how many heads it's supposed to have. And what we can do is we can take this animal and we can, in place, rewrite that electrical pattern so that now it says 2 heads. And when you take this animal and you injure it, the cells obey this pattern and they make a 1-headed animal 100% of the time. It's very consistent. But we found out that if you rewrite this pattern and then you recut him after that, what they will actually do is consult this pattern and happily enough make a 2-headed animal. Again, not Photoshop. This is real. So this is a kind of counterfactual representational memory, which is that this body is representing what it is to be a correct planarian, meaning what is my target state in this anatomical morphospace. And that memory is latent until you injure it, and then it does its thing. I call it a memory because if you amputate these heads and let them do their thing in plain water, they will continue. So it's rewritable, right? The memory is it's long-term stable, but it's also rewritable. And this is what these 2-headed worms look like. So there's nothing genetically wrong here. We didn't change the hardware specification. All the cells have the same proteins. We didn't touch any of that. It's the large-scale goals that we've altered for this collective intelligence. We've now redirected where it thinks is the right direction to go in this anatomical morphospace. Not only can we do that with a number of heads, but we can do that with the shape of heads. So we can make this guy create the heads of other species. So flat-headed P. felina, round-headed S. mediterranea, without any genetic change at all, just by modifying the bioelectric states that serve as the guidepost to its movements in morphospace. And so that same piece of hardware can visit other attractors in this morphospace that typically belong to these other species. And it's not just for the head shape, but for the shape of the brain inside, the distribution of stem cells, exactly like these other species, 150 million years distant. And so, just kind of the last biology example I want to show you to really nail down this idea of a collective intelligence is this: using the same bioelectric techniques, we can modify in an early frog embryo where the different organs are supposed to be placed. And so we can convince a bunch of gut cells that they need to make an eye. And they make a complete eye with all of the same lens, retina, all the same stuff. And the way we do that, we don't specify all the details of how to make an eye. We don't program it by dealing with stem cells or telling individual cells how to differentiate. None of that. What we found is a high-level subroutine call that basically says to these cells, build an eye right here. And that subroutine call is a particular bioelectrical state that we can induce by putting in some ion channel RNA. But so it's very modular. It's top-down control. It pushes all the complexity onto the system. We don't need to know how to make an eye. One of the coolest things is this is a lens sitting out in the flank of in the tail of a tadpole somewhere, and you can see the blue cells are the ones that we manipulated with our ion channel. All of these other cells are ones that we never touched. And so much like recruitment in ants and termites and things like this, these cells, having been given the task to make an eye, can also do the computation of the fact that there aren't enough of them to make an eye. And so what they do is they hack their neighbors and they re-specify them, and they say, look, you were going to make some tail skin, but instead of that, make a lens, make a and make some other stuff, because there's not enough of us to do this alone. And so the secondary instruction, right? We instruct these cells, they instruct their neighbors. We didn't have to program any of that in. That is just completely native to the biology. So this collective intelligence, much like other collective intelligences, has lots of competencies. It scales to the appropriate task. It hacks each other as needed, and so on. And so this allows us to have a different different look onto this whole issue of the Xenobots. Typically speaking, if you ask what did the Xenopus laevis genome learn during evolution? Well, it learned this. It learned a series of developmental stages, and then some tadpoles with tadpole behaviors and so on. But and so that's what we see if we just look at standard behavior. But if you perturb the system and liberate the cells from their normal instructive interactions, where these other cells are typically basically bullying these skin cells into having this boring two-dimensional life on the outer surface of the animal, where all they do is get to keep out the bacteria. Left to their own devices, this is what these cells actually want to do. This is their native behavior when they're not being behavior shaped by other cells. They make a Xenobot. They make these crazy developmental sequences. I have no idea what this is. Nobody's seen this before. They have their own behaviors. And so what evolution actually did was not just to make a thing that's perfectly fit to a froggy environment. It actually makes a problem-solving machine that, due to its multi-scale nature, due to its this competency architecture where all the pieces have a collective intelligence that solve problems, it's competent to do many other things. And that means that, unlike every other animal you've ever seen, when the question is, why does it have certain behaviors, a certain shape, certain structure? And the answer is, well, selection. Eons of evolutionary pressure is shaped at this and that. There's never been any Xenobots. There's never been any selection to be a good Xenobot. All of this is emergent from the fact, and this is, I think, is maybe one of the most interesting things out there, that evolution makes these amazing problem-solving machines with novel behaviors. And so I like to think about it this way. This is kind of an homage to Doug Hofstadter in his famous book. This idea that that we now have the tools of computation, of cognitive science, and of basal cognition to really think about evolution providing us with a kind of hardware that you can look in different ways through this tool, through this lens, and you can see different shapes and different aspects of morphospace that are completely not obvious, the exact same hardware. So I'm going to end here just by summarizing the main points. I think that biological life is a set of overlapping collective intelligences of these multiscale agents. I've got this TAME, T-A-M-E, stands for technological approach to mind everywhere. This TAME framework, which has all kinds of implications for regenerative medicine and robotics and so on. And I think that these biological robots are a tool to explore new spaces that we really are not very good at predicting, and we really have to raise our game in terms of recognizing and relating to radically different composite beings, because biology is so interoperable. Pretty much any combination of evolved material, design material, and software is some kind of agent. So we have hybrids and cyborgs and all these things. Many of these already exist, but increasingly, in your lifetimes, we're going to be surrounded by things like this that are nowhere on the tree of life with us. So all of Darwin's endless forms most beautiful are like a tiny corner of this incredible option space of bodies and minds. These things are going to be everywhere, and we really need novel frameworks to be able to ethically relate to beings that are really quite alien. So that's it. If anybody's interested in the details, here are some papers where we take this apart in considerable detail. And I want to thank the Xenobot team in particular, Josh and Doug and Sam, and the folks that worked on the eyes and the planaria, and I thank our collaborators and our funders, and especially the model systems, because the animals do all the heavy lifting here. So thank you very much, and I'll take questions.